great crowd. It was great this morning. It's still great now. I apologize that Ron Sullivan is not here to introduce Greg Hampikian, but I am thrilled to do so. You've got his bio in your program, but he's a professor of biology and criminal justice from Boise State University and founder and director of the Idaho Innocence Project, and has been involved in forensic DNA testimony and cases like those of Amanda Knox and Sarah Pierce that you, I'm sure, will have heard of. He is a true Renaissance man. This is quite a bio. Also an award-winning playwright and a fellow of the National Academy of Inventors. He holds patents for power generation and a miniature pump using magnetic shape memory alloys, whatever that is. This cannot help to be a fascinating talk. So Greg, please welcome.
Yay. First of all, happy Wrongful Convictions Day. That's honestly, that's what today is. It's the first time the International Innocence Network has decided to declare that day. And another coincidence is that 4 years ago today, I was at my home in Boise, actually just flying out to CNN in Atlanta because we were going to hear about Amanda Knox's appeal. So she had lost at trial. She had been incarcerated for 4 years in Italy. At that point, I'd been working on the case for 3 years, I think. And we were going to hear if the judge would free her. And I just saw her sister just posted on Facebook that tomorrow's the anniversary of that release. So it's great to be here to talk a little bit about DNA, and especially something that I do a lot of now, which is post-conviction testing. That is, people who claim innocence and write to us. I work at the Idaho Innocence Project. I volunteer as the director over there, but I helped start the one in Ireland, work with the Georgia one, and really all over the world now. Because I'm the only scientist who's the director of one of these organizations, they send me all the really hard cases, hopeless ones that take a long time to solve, and I enjoy that work quite a bit. So I'm going to talk about some advances in DNA, and I'm going to talk about some problems with DNA, because I'm a bit of a person who likes to hunt down problems. Last time I was at Harvard, I think this was the last time, I was here for an Innocence Network meeting, and the three gentlemen in the front here are three men whose cases I worked on for the Georgia Innocence Project, just to show you that it doesn't all stay in the lab. One of the people in this picture actually was a student and helped free two of them as his internship. Most people who volunteer with us leave very disappointed because, as I say, these cases can take 10 years and they want to stick around for 6 months. People always ask about death row, and Charles Fain from my state, I did not work on his case, but he spent 18 years on death row for the rape and murder of a little girl near where I live, and DNA got him out, mitochondrial DNA got him out of prison. But if we speedily dispatched the justice system, people do tire of justice taking so long. Charles Fain is not one of those people. He would have been killed. The average time on death row is over 9 years before someone is exonerated. So a lot of these people that are being killed, of course, the network groups do not get involved with their cases. We don't know how many innocent people have been killed. So I want to show you one of the great things that DNA can do, and one of the things that we don't do so well. So I was talking with some computer science people last night at supper, and they said, one of the things people do better than machines is facial recognition still. And that's true up until this point, though there's been a lot of improvement. So this is a standard lineup. This is actually a lineup that John White, here in the center, was called for. He was working. They said, can you come in? There's been a rape. A woman, an older woman who was 74, had been raped in the neighborhood and beaten. And they said, look, we can just get this over quickly. She's going to go through the lineup. He said, sure, came in. They're like, where do you want to stand, John? He's like, right in the middle, let her get a good look. And she picked him. And so that was 1980. The student that I showed you in the first slide worked on this case. And we actually got John White out just a few years ago. And when we got him out, we got a hit to the database of the actual rapist, or whoever left the semen on this woman who was raped. And we were very excited. We got the name, and the student said, I've seen that name. And I thought, well, a lot of people write us, and some of those people are guilty. So maybe this guy wrote us from prison. He was in the system. That's why his DNA was in the system. And he said, no, I know it's from somewhere. And so he went through all the stacks of information that we had in this case and found out that James Edward Parham, by absolute coincidence, happened to be there at the jail that day. They stuck him in a lineup. They're like, you're just a filler. You know, if she picks you, don't worry about it. You're not a suspect. That's how bad we are at facial recognition, even though we're better than the computers. Okay. So DNA cleared this up, and so I want to say, yay, DNA is great. And then I'll talk about some of the problems. So imagine that we can find a gene that causes crime, and we could just eliminate that gene from the gene pool somehow. That's kind of a part of what I posited in a piece I wrote for the New York Times that was titled by them, Men Who Needs Them, which got me hate mail from men's groups. And I was like, where were you when I was an angry divorced man? You know, anyway, these are mostly guys who've lost custody battles who were very upset. There was only one I turned over to the police, but I got hundreds of these letters. And I was just talking about the Y chromosome and the fact that, you know, most of my clients in prison for violent crimes are men. And so we have the gene. Well, we know where it is. It's on the Y chromosome if there's a gene. Should we do anything about it? But mostly I was extolling the virtues of the female half of our species, which I consider the only living half. I think the men, you know, we only give 3 picograms to that baby. She's losing bone mass. And if you added up all the mass of the male contribution to humanity from the beginning, all these millions of years we're talking about, it's less than a teaspoon. So anyway, I recommend you take a look at that if you want. But the way I got into forensics was I was working on the Y chromosome in marsupials. So I titled this DNA is Universal from Fruit Fly Eye Color, which I worked at University of Connecticut down the road for my PhD, as part of my PhD, to murder and rape. And marsupial sex determination I threw in there because that's what I did in between. I went to Australia, thank you very much taxpayers, on a National Science Foundation grant to study marsupial sex determination, and was part of the group that found SRY, the sex determining region. Our collaborators in London turned a female blastocyst mouse into a male mouse, and that allowed me to give talks all over the world. It was a lot of fun. Then I became a house husband after that for a while to take care of our kids, so I learned a little bit about what a lot of women go through. But because of the Y chromosome work, I started getting calls, could we identify a stain as male or female if it was spit? Now, this was 1990, I thought, well, yeah, we could do that using SRY, or blood, or anything else. And so I started consulting a little bit in crime in the early 90s. And just to show you how universal all this is, the eye color of Drosophila, this is a kit developed, the Irisplex system by Manfred Kaiser and others in his group, that now we can look at a stain because of their work and others and tell what eye color it is if it's light blue and if it's dark brown. Now, the hazel people, you give us a little bit of a problem. And to show you just how sensitive all these tests are, it's these single loci, these single areas of the genome where just single base changes can make changes in eye color that are predictable. And so we used to go to all the forensic meetings, and they'd scan our eyes, and they'd take our DNA, and they built this database, and apparently it's very good. I have not seen this used in court or in any crime solving yet, but it's a pretty robust kit and has been reviewed over and over. So my lab got involved with some of the newer technology as well. My mother lived in Paris, and so I got to know some gendarmes and as young folk, and then they went on and became fairly important in the police departments. And they called me about a case that had languished for 10 years where a woman was, she made a 911 call, and all you can hear is her screams, and then a couple of men. And then they found her body burned, but some semen was recovered. They got a full hit. I mean, they got a full profile, right? Every bit of the DNA that they wanted to measure, they measured, put it into the French database, no hits. So no criminal was already in the database. And they called me up and said, look, we've heard something about familial testing. Nobody's tried it in continental Europe. They've done it in England. Do you think we could do it here? We don't have the software. And so I hooked them up with some friends who had the software. It got all the way up to the president's level in France, and the president said, no, we are not going to do familial testing in France. And so I said, well, there's one other trick. And if you just set the dial on the match stringency that the FBI software has, the CODIS, you could just set it for a half match, basically. Don't find me a perfect match. Find me half of the chromosome markers that are in common. Well, who would that be? Dad, son, right. And so that's what they did. And I thought, you know, you got 1,800,000 in the database. I had no idea. I'm not very good with the math. You know, I don't know how many hits they were going to get, but I thought we'd get at least a few dozen. Turned out we got just one hit. So this is just part of what the crime was, the facts of the crime. They had a condom with some semen recovered, cigarette butts, nothing for 10 years. 1,800,000 convicted offenders and arrestees, and we got one hit. We then confirmed that with a male-only Y chromosome test, then got a sample from the putative mother, found out all the missing alleles were there, and found out that the person that it matched as a potential would be a son of this guy. And he had a son about the right age who had died, Gregory Weert, he died in 2003. And so before they exhumed the body, I'm guessing that they tapped all the phones and found out when they started digging up the body who was making phone calls among his old friends. They got a voice print or a voice expert to identify the voice of one of the friends, and they think that was the second voice on the 9/11 call. So that's the way these things are starting to evolve. And even if you are not in a database, right, even if you're not in Family Tree or whatever, eventually we'll be able to find you. We'll just reconstruct the missing bits. And for the purposes of identification, we will probably be able to find you. Right now, we need to confirm it with actual tests. Something new that we got involved with, I have a very similar case in Idaho. Chris Tapp is a man who was interrogated for 35 hours. I think, was it Dateline who did the show on him? One of the big shows came in and did a story on him anyway. And after all those hours, they offered him a deal if he would just say who the other guy was, because the DNA didn't match Chris, but they thought maybe he knew who it was. If he would just say who the other guy was, they would give him no time in prison. And so they'd been interrogating him for 35 hours, telling him the name of this guy. And so he gave them the name. They tested the guy, and it wasn't a match. They came back. They said, Chris, it wasn't him. Who else was it? So he mentioned everybody they mentioned in this small town during the interrogation. They tested 54 people, I think. Chris was crying at the end saying, I'll give you my mother if you think it's her. Who do you think it is? And anyway, so they revoked his deal and charged him with murder and rape, even though the DNA on the semen on a dead girl's body didn't match him. And we've tested things since then. Pubic hairs, we've had them test that were found on her face, et cetera, et cetera. Clothing that they tested, everything matches one guy, not in the Idaho criminal database. So working with the police, we suggested, what about looking for the last name of the semen? Treat the semen like an adopted child looking for its daddy. And so they did that. I think they used methods that maybe I wouldn't have used, and it's been a bit controversial. But they did some genealogy testing, got the last name of someone, tested that person, it wasn't him. And that shows you the problem we then have. We have to go to the paperwork, and find out who else is related to that Y chromosome. But that's the kind of thing that's going to happen. And I think it was mentioned if everybody was in the database eventually, how much easier all this stuff would be. Okay, this is kind of the results you get when you do that type of search of publicly available databases, but sometimes you get people who don't want their name listed, and so you won't get a last name, and there was a court order was used in that case, in the case I talked about. Okay, some basic DNA. Let's look at how it's done just really quickly. Here's this cell. Hopefully you've seen one before. That's the nucleus. This is a gigantic mitochondria. They're not that big. There could be 1,000 of them in the cell. We get them only from mommy. Like I said, mommy gives us everything, right? Daddy gave us a little bit of sperm, and he's actually not even there when the nucleus enters the egg. You know, he's already gone. It happens a day later or so. But anyway, the mitochondria come only from mom. There's thousands of them, so are thousands sometimes of them. So we can even get mitochondrial DNA from a broken hair. Mitochondrial DNA was famously used to identify the unknown in the tomb of unknown from the Vietnam War. So there is no unknown from the Vietnam War, and the family had to fight to get that done. The military resisted. But anyway, that tomb is now not only unknown, it is empty. We get our DNA from swabs. I carry them in my bag wherever I go. And basically, it's a Q-tip type of thing, and the cells look like this unless they're sperm cells. Sperm cells are really tough. They have a disulfide head, and so we can separate them from all the other cells. It's the only type of cell we can separate, and so that's how in a sexual assault we can get what we call the male profile, all the male contributors. And we don't care about the 3 billion bases. We only want to know the size of 13 areas. How big a piece you got in the sperm, how big a piece you got in the egg. So for several of the chromosomes, we do that. If we have 13 of those areas, just the size from mom, the size from dad, and they're numbers like 5, 7, very, very simple numbers. We know those sizes, we can find you. So there's the cell, we magnify it, I don't know, a few hundred times, again, a few hundred more times. If we could see the bases, it'd be like this. And we're just going to amplify this with something called polymerase chain reaction that won the Nobel Prize for Kary Mullis. Here are the 13 areas that the FBI currently requires. They're going to be expanding soon to over 20. But these are the chromosomes. And chromosomes are nice because they were discovered before anyone really knew how they were working very well. And so the big one is called 1. The less bigger one, 2. There's 1 actually kind of out of order. But, and then you have the X and Y here. Is this a man or a woman? It's a man, right? Y chromosome. So if we have a Y chromosome for humans, we're generally a man, unless you have a mutation in SRY or a couple of other genes. All right, here's the data. Ready? I want to teach you how to read one of these. This is called an electropherogram. Can you say electropherogram? Electropherogram. Great. You're on your way to testifying. So now, those areas, those 13 areas, this is one of them right here. This is from chromosome 3. And you see the sizes, 16 and 17. So mom and dad had 2 different sizes that they gave this person. This is another location on a chromosome. Mom and dad gave the same size there, 15. And that's why the peak is higher. It means there's more DNA. Here's another one, size 21, 22. When I find then the frequency of the 16 allele in the population, I can multiply it by the 17 allele, and I can find out how often I would expect this profile to come up in the population. That's the statistic I would use. All right, so this is a single contributor, no more than 2 peaks at any one location. Now let's talk about some of the problems. I'm very concerned about the problems in DNA because I think we're having a large number of wrongful prosecutions with DNA mixtures, where there are mixtures. Or where there's very small amounts of DNA, like in Amanda Knox's case. And she was convicted based on a very small amount of DNA that they found on a knife blade that was consistent with the victim, and Amanda's DNA was on the handle. We said that DNA on the knife blade was spurious, probably a contaminant. It was never repeated. It was below the level of DNA the FBI would even look at the time, or any of the labs I worked with. And that lab the Italian lab hadn't been accredited yet, certainly not to work with that low template DNA. So just because there's DNA doesn't mean that it's good science. Report language can be very weird and be very biased. For example, you can call something a profile when there are several people in it. So you can say the profile matches the client, but there's actually several people in the profile. And oftentimes that lack of a plural confuses members of the jury. And then we can talk about statistics and DNA transfer if there's time. I'll get through as much of this as I can. OK. Peter Gill, one of the great experts in our field, has said, look, if you show 10 colleagues a DNA mixture, you'll probably end up with 10 different answers. You won't hear any experts say that on the stand who's coming in for the state, though. They'll just say, this was a match, here's my calculation. But all of us in the field know that this is a problem. We didn't know how big a problem until Ideal Dror and myself did a study, and now John Butler at NIST has followed up, and I think they're very telling, and I'll show you what they are. So this is what a mixture looks like. OK, so in any case, there's lots of peaks here. And there's 3 peaks here, so it's obviously more than one person. So what happened was, with that data from that slide, we took that data, So an actual case where there's a man in prison, Kerry Robinson in Georgia. I believe he's innocent. He was convicted of being in a gang rape. A guy whose DNA was perfectly in that sample from that poor girl, and who admitted to shooting people and to raping her, then was offered a deal. Tell us who the other guy was. We'll knock 8, 10 years off your sentence, whatever it was. So he said, Kerry, and he gave a wrong last name. And then he said, and they're like, oh, Kerry Robinson. And so they went and arrested Kerry Robinson, a guy he didn't like. And Kerry's like, I didn't do it. All they have is evidence for Kerry Robinson. The woman who was raped picked the other guy out, didn't pick Kerry out. All they had for evidence was the word of this guy who was making a deal and a corroboration of the DNA scientist who said, I can't exclude Kerry Robinson from that mixture. I think he's excluded. Eric Carita, who was at the Connecticut Crime Lab, thought he was excluded. And Fox News in Atlanta did a story on it. And I said, look, this is a statistics problem, but I think we can explain it. I said, swab 4 people. Here are swabs. Four of your reporters, swab them. They swabbed them. I did the DNA. All of them were included, according to the GBI. So we took that data and we gave it to 17 scientists at an accredited North American crime lab and asked them, is this profile included, excluded, or you cannot conclude is inconclusive. We gave them no contextual information, and most importantly, we didn't allow them to talk. Like if I allowed you to talk, you might all decide I'm giving a good talk, right? But there's 15 of you in here who think I'm terrible, right? We do this group think, maybe 10. We do this group think, and we come to conclusions. Oftentimes, you do a straw poll of a jury, they find a client innocent. By the end of the day, that's their first straw poll, by the end of the day, they can convict. We process things socially different than we do when we're alone. And so we did the study, and out of those 17, only one scientist at that lab agreed with the Georgia Bureau of Investigation. Only one of the 17, and we published it in a peer-reviewed journal, Science and Justice, and it got a lot of press. So The Economist picked up on it, New Scientist picked up on it. 1 out of 17, I'll just skip that. So then I had a lawyer write the head of the Georgia Bureau of Investigation's DNA, and like, what rules are you actually applying? Because it seemed like even if you had one allele in common with all of those markers, just one, and I have one in just about every case I'm with, they would say you cannot be excluded. And in fact, what the head, you can't read this, but what George Herron said was, even if there are no alleles in the evidentiary sample consistent with or matching those observed in the subject's known DNA profile, no alleles with the evidence, this does not constitute a conclusive exclusion of the subject, since the absence of evidence is not the equivalent of the evidence of absence. What this means in practical terms is, anybody who wants to reduce their sentence could point to any of you in the room, and if you had 0 DNA in common with the, with the evidence, that would be a corroborating fact because you cannot be excluded, right? That's all it takes. So while I can understand how George might have said this, like what he's trying to say is I can't say, he should have said it's indeterminate, right? It's inconclusive, not it's cannot exclude, which in DNA language he should know. In DNA language means you're included. Okay. There were no stats used in the report that convicted Kerry Robinson. Thank goodness, 2010, the Scientific Working Group on DNA Analysis and Methodology says you have to have statistics. Hooray, we all say. You know, now there's going to be science. Okay. So John Butler at the National Institute of Standards and Technology does something like what we did. He starts this study in 2005, actually before we did, and just gives the same data to a bunch of volunteer crime labs all over the country. They pick out the right people, and then he asks them for a stat. Give me a match statistic. Now, when you go to court, you have damage to your bumper, right? And you have to have 2 receipts generally, right? One for $30, one maybe for $45. The judge chooses the one in the middle. With DNA, he gave the same DNA, and what did the stats look like? These are the stats for one of the samples, and the conclusion was there was some of the labs had it at 10 to the 5, and some had it at 10 to the 15. There were 10 orders of magnitude. That's the difference between a $30 damage to your bumper and a $300 billion damage, right? So you should get, what, the $150 billion, right? Minus, give or take, 30. So that was 2005. 2013, you know, we've published our paper. I thought, oh, you know, finally they're going to do something about these mixtures. I've been talking with John Butler at NIST and asking him to redo this study, please, please, please. He always intended to. And please, please, please publish your last study. He never did. I'm hoping he'll publish this one. And they had 5 different cases in 2013. They sent the same data out to all these volunteer labs. So 4-person mixture, 4 people from John's lab add DNA to this thing, and they pretend it's a ski mask, send the data out to all these labs. They also send the labs 2 of the lab people's profile who were actually in the mixture, and 1 person who wasn't. And they say, who's in, who's out, count, give us a stat. 108 labs. C is the person who is not in there. Out of 108 labs, only 7 excluded C. You could go to prison for the rest of your life on a mixture, okay? And this is the kind of nonsense that's actually going on in this country and all over the world. This is not science the way it's supposed to be done. And DNA got a pass when the National Academy of Sciences looked at all of the forensic sciences. They didn't like bite marks. They criticized fingerprints because they don't have a database. They love DNA. And I was just very upset at that day when that came out. So out of those labs, 76 of the 108 included the person who wasn't in it and provided a match statistic. The match statistics weren't like 1 in 10. You know, it could be anybody. It was 1 in 9 for some labs, could be anybody, to 1 in 344,000 that I guarantee you would convict. All right. Where else does this happen? This is Taiwan Association of Innocence, a new organization, came to one of the national meetings in 2013. They were telling me about this guy who was arrested because there were 2 escorts who had been raped in a warehouse, and this guy had an interest in the warehouse. There were 3 suspects. 2 of them are convicted in 2010, so a year later. And then they start prosecuting Mr. Chen also, who says he was innocent, never had consensual sex. The other guy said they had consensual sex with the women. So here's the Y chromosome profile from the underwear. Okay, this is the victim's underwear. This is a typical way we work a case. These are just locations on the Y chromosome and what size DNA each person has. So that's Mr. Chen, suspect 1, suspect 2 who were convicted. And as you see, at the first location, Chen is a 15. What's the underwear? 15, so it's a match. Anytime I say match, my students have to say statistic, right? So match, statistic. That's probably, actually we can't calculate it for this, but that's probably, let's say, a 1 in 10 for the purpose I'm gonna put here. A 12 allele, they're all 12s. Well, there's 12 in the underwear. We go through, guess what? Poor Mr. Chen, he's in the underwear, right? It's a match. That's now statistically pretty robust, right? We have all of those parts of the Y chromosome. In fact, that's the entire kit. We can't go any further. What do we say? I say to the people, why won't we see if we can do more locations in the Y chromosome? Maybe he'll be excluded later. So they go to court, and more of this date. So he's guilty in 2012. His appeal's denied 2013. We take the case 2013. And we go to court, and the Taiwan lab worker said, we have a new test. It's got more loci. It's got 6 new locations in addition to the other ones that were already there. And there's Mr. Chen. He's a 17. He's excluded at 2 of the locations. There's Mr. Chen. And finally, I got to meet Mr. Chen because he could travel just a few months ago. So here's again a case where you have a DNA match. It looks good, it looked good even to me. The full kit was used, but it's a mixture. And these mixtures, in reality, are very difficult, and they are confounding juries. Now, I told you about the mitochondria, and mitochondria are good for us because if there's a fraction of a hair left without a root, we can use mitochondrial DNA. We do a lot of mitochondrial DNA on the Basque. I study the Basque in my lab as well, and we have some projects on them. But anyway, the mitochondrial DNA is something you get from your mother. We can do lineage stuff, and it can be used when there's just a fragment of hair or the body's very decayed or burned. And Donald Denman, this is from the Albuquerque Journal, he went missing. He was 49 years old. His family's troubled. They never had the body, never found anything. Finally, some bones are found near the house. And so they identify those bones. This is February of 2008, and donations could be sent to the Salvation Army for Donnie Denman. The FBI confirms the DNA by mitochondria of those bones. Since it's from the mother, they get one of the mom's relatives, one of her sisters in another state. They finally confirm it. Now we're in March of 2008, same year. It's tough for his brother to look at the photograph of his brother as he stares at the image. He couldn't help but wonder, would his brother have turned his life around if he had lived? This is now May 3rd, same year. I said match. You didn't say statistics. So with mitochondria, it's generally 1 in 2,500, 1 in 5,000, something like that. And so it was a coincidental match to somebody else's bones. A friend of Donnie Denman, who's repairing his car in his garage, putting down newspaper, sees the obituary, calls Donnie and says, Donnie, they buried you. And he's like, you're kidding. He's like, well, who spoke at my funeral? Like, your pastor. So he called the pastor. And like he says, I'm very much alive. It's a trip, isn't it? Okay. Once again, good DNA, a true match, a statistic that is not an identity. It is a good statistic. I mean, 1 in 2,000 sounds good. 1 in 5,000 sounds good. So now we'll talk about sensitivity. Can we become too sensitive? Here is a slide of some sperm. When I was first doing DNA in the 80s, we'd need about 500 of these slides to give a really good DNA profile. We're using a technique where we couldn't amplify DNA yet because it hadn't been invented. Now, single cell. All right, so I want you to consider this. As our instruments and our kits have gotten like a millionfold better, yet most police work is still collecting evidence the same way. Let's think about what a problem might be. So I try to boil this down to people's practical experience. How many of you swim in a public pool? Have swum in a public pool? Great. So the problem is this. What if one person, man or woman, goes into that public pool with an ejaculate on them or in them, right? And I got news for you. As you're swimming, that water is doing more than going over your body, right? It's cleaning you out a bit. And if one person goes in, 300 million sperm cells from one ejaculate in that public pool, how much water do you have to swim through before you get a sperm cell? Well, there's 300,000 gallons in an Olympic pool. It's pretty easy. There's 1,000 sperm cells per gallon for every ejaculate evenly dispersed. Now, if you happen to hit a rich area of ejaculation, swim with your mouth closed is what I... Okay, so in 1982, I'd need 1,000 of those gallons to do a DNA profile. Today, I need about a teaspoon. So how does that work into forensic casework? Well, Meredith Kircher was the young lady who was killed in Italy, the student who was killed that Amanda Knox served time for and was eventually exonerated for. That's Amanda and Raphael, her co-conspirator who supposedly were involved in the murder. They were suspects before any of the DNA from the room or the fingerprint evidence was done. And so it was a gut feeling from the prosecutor. And can you have gut feelings in science? Yeah, they're called hypotheses, right? But the difference between a theologian, some of my friends who went into theology, and me who went into science is, you know, a theologian will die for their ideas, and that is the measure of their dedication. And a scientist's measure of dedication is you let your ideas die. You put that baby that you, I mean, I've tried to cure cancer for a long time. You put it up for a test every time, and everything you thought was going right, you abandon. There was a lot of press on this. They spray a tan on you at CNN. That was before the beard. So only 2 people's DNA, when they finally do the DNA and the fingerprints, only 2 people are in the murder room, the victim and Rudy Gaudet, who they try and they put in prison, and they still persist with Amanda and Raphael based on that gut feeling. Finally, we had the ruling from the Supreme Court just a few weeks ago that said, no, they got it completely wrong. She's completely innocent. It was ridiculous. She should have been released immediately. He's serving time. This is the bra clasp that convicted Raphael, Amanda's boyfriend of a couple of weeks. They picked it up 46 days after the murder, after the teams had been through a couple times. They said they forgot it. I could show you the collection film. They actually have it. They're passing it around. They're touching it, passing it from one guy to another, and then they put it back on the ground because they forgot to photograph it. So here they are, like totally in the bunny suits, and they're putting evidence on the ground. Anyway, and they said they're changing their gloves, and their gloves were clean, and we had evidence their gloves aren't clean, and we showed all that video. That's the knife that supposedly Amanda used. And that's where DNA consistent with the victim was found. This is the electropherogram of that DNA from the victim. What is important here is that the y-axis, which the FBI at that time wouldn't look at anything below 200, there's only one peak above 100 here. They were never able to repeat this. It was an invalid procedure, and that's why she's free. We repeated this in my lab as we were studying it. We collected soda cans from the dean's office. at lunch. And then I told my people, save money on the gloves, just like they apparently did. Don't change your gloves. Change it every other piece of evidence. Collect a can, go into another room, and unpack a knife from the dollar store and put that in a separate evidence bag. Keep everything separate, then change your gloves. Do it for 5 people. We did it for 5 people, and we found one person's DNA transferred to the knife, 1 out of the 5. I'm going to skip through this just because we're at the end. Whoops. So happy ending. There we go. Amanda, that's a few weeks after she got out. We had Thanksgiving together at her house. And then both of our families went rafting for 3 days in Idaho. I'll show you one more example of how this can go. And somebody's going to have to shoot me to get me off the stage. I mean a photograph. My mom lived in Europe, so I followed this story for years. For 15 years, this woman was committing crimes all over Europe. Rarely was there a witness. There's a murder. There are Romani chiefs feuding gypsies who use guns, and her DNA is found on the guns. She robs, she injects herself with heroin. They found syringes with her DNA. Kills with almost professional precision. As far as German detectives are concerned, she has no identity. They test 800 women in Heidelberg. They do DNA tests on her. Nothing, no match. Bruno Bosch says in 2008 he's traveled 60,000 kilometers questioning witnesses. Nothing. They did 800 women, as I told you. And in the end, it was a woman who worked in the Bavarian swab factory. She was leaving a little something on her swabs. That's the phantom of Heidelberg. Why wasn't this detected? Because we don't track our errors in forensic science. And I'll show you my lab's answer to that. We study the smallest sequences that don't exist in nature. We call them nullimers. For DNA, they were originally 13 bases long. For proteins, they were 5 amino acids long. And we call them nullimers, and we've made cancer-killing agents out of the proteins. But out of the DNA, what we've done is we've made a marker that amplifies better than any of the human markers. And it looks like this on an electropherogram. Those are human markers that have been diluted 100,000 times after PCR. And then we diluted it 1,000,000 times and tried to PCR up everything, all the human markers and our nullimers, put it on a knife, and you can still see that. And so that means when you give a swab to the police, at least one thing we can avoid is they make 1 billion copies. That's what I do in my lab. You give me DNA, I make 1 billion copies. First thing I do. Each of those copies is indistinguishable from the original, right, in PCR. It's not like a traditional fingerprint. The Supreme Court got that wrong when they said that it's just like a traditional fingerprint. We can take them from arrestees. It's not. I make 1 billion copies, and one of those copies in an aerosol on a tube and a glove is enough to convict you, and that's why we've invented the nullimer marker. So I'll skip all this. If you're interested more in hearing, because we're out of time, right? You can read my book or contact me at hampikian@yahoo.com. Thanks.
Yeah. We've got time for about 4 or 5 questions. If you guys want to come, is the microphone out? I can't quite see. We've got a microphone. I guess we can, in the middle there. Come on up, say who you are, and if you can just keep it to a question, that would be awesome. And maybe while we're waiting for folks to come up, I'll just ask you. So, if you were to recommend one thing to the police, one thing to the labs, one thing to the analysts to improve this system, what would it be?
So what I tell my students in lab is keep your arsenic out of the spice cabinet, right? And for God's sakes, if you keep them together, don't keep your arsenic in an old spice bottle. So what I'd say is these are the, I'm going to do it by demo. We work in very small volumes in lab now, and these are the racks. And you can't possibly read the writing on the little tubes in here, so it's all about the order they're in. I skipped a case. I have 2 cases where they were just swapped tubes. That's what put people in prison. And Las Vegas just apologized to a guy they did that for. So I would say keep suspect things separate. They should go to a separate lab. There should be one national lab, or maybe a couple national labs that just do known samples. And then the rest of the crime labs do their thing. But they've got to keep them separate all the time. I think there's a lot of mix-up being done, and the tragic thing about those is there's no way to tell. There's nothing I can do for somebody if that's happened.
Great, we've got some questions.
Yeah.
Hi.
Hi.
Great talk.
Oh, thank you.
Given that we know that there are trace amounts of genomic DNA all over the place, and it's quite hard to amplify STRs and stuff like that from trace amounts, what do you think the probability is that the forensics community will turn away from STRs and mitochondria into like low coverage genome sequencing?
Well, it's funny you should ask. So the FSI genetics that I just picked up in the mail on my way over is New Trends in Forensics Genetics. And it's all about massively parallel sequencing, even things like microbial genetics. Do your microbes identify you? Probably. I want to do a study actually about people who take antibiotics and then get divorced. Is because after years we develop the same microbiota, right? And if one partner is taking antibiotics and going traveling, you change your fingerprint. Does it change your behavior? Does it change your interest? Do you find a different smell pleasant? So they're looking at microbes. That's all free stuff. That's my idea. You can take it and run with it. But they're looking at other ways of mostly this massively parallel sequencing. You can now, with mixtures, I can only tell 3 people apart. They can look at 1,000 people and say if one person is included or not. So yeah, it's pretty impressive, but nobody's used it in court. Yeah.
Hi, I'm Denise McWilliams. I'm with the New England Innocence Project, and thank you for your help to us over the years.
Yeah, you're welcome.
I recently was at a conference in New York where I watched the scientists and attorneys for the prosecutor and the defense literally scream at each other about the use of low-copy DNA. Mm-hmm. And I'm just
that was in New York?
That was in New York.
Yeah, of course.
Just curious what your thoughts are about the use of low-copy DNA.
Well, so low copy means 2 different things. So the original meaning of that meant we did extra rounds of PCR. And in that case, you actually see what we call drop-in. And what all the drop-in is DNA that's just kind of floating around or in the glove when you buy, on the glove or in the tube, that you don't see with these normal 28 rounds or 30 rounds. But when you do the extra rounds, you get drop-in. But low template DNA, which probably is something they were arguing about, is when you have very low levels, like in Amanda Knox's case, you'll get different results in some of those cases. And so in New York at the OCME, they were saying, if you get the result 2 out of 3 times, you can report it. And when I did a case in London, they said, well, it has to be repeatable. And I thought, well, that makes sense. A repeatable result is reliable. And then I looked at their records. They did like 10 tests. 2 of them were what they wanted. They picked the 2. They called it repeatable. So I think there's a lot of problems with some of those techniques. But frankly, that bar keeps moving. As we get down to single-cell resolution, it won't be as much of a question anymore. So we keep lowering the bar of what low template is. So yes, it should be used. Yes, there are problems.
Hi, I'm Faye Flam. I'm a journalist with MIT Technology Review, and I was intrigued by these cases where there were mixtures and the statistics looked good, where there was like a 1 in 2,000 chance that it was wrong. Is the problem there that the person was the 1 in 2,000, or the problem that people are screwing up the numbers, that they're making statistical errors?
The problem is that there are several good methods to do statistics, and they produce extremely variable results. So when I say good, they're based on mathematics. All the statisticians agree they're good, robust methods, but you get 10 to the 10 different differences. And then you get labs that will exclude. Most labs are still applying kind of rules of thumb. If there's only 1 allele, we won't call it. Utah told me there had to be at least 2 that weren't found to exclude somebody. They all have these kind of rules of thumb that they're applying. So the statistics are helpful, but also I think our idea about these statistics is wrong. If I say 1 in 5,000 in a Boston crime, that means there's 1,000 people in Boston who are a match, a definite match, right? And so if you think of it in terms of epidemiology or in terms of medical testing, that's a false positive rate of 999 to 1. So for every 1 person in a 1 in 5,000 match in Boston, there are 900, for every right one, there are 999 wrong ones, right? And it doesn't mean we don't use that, but you have to start giving a better perspective, because jurors hear 1 in 1,000, 1 in 10,000, 1 in a million, and they think it's over. That's, they don't hear numbers like that. They hear 50-50. It's 20% chance. When they hear those kind of overwhelming numbers, they generally convict.
Thank you.
Yeah. Okay, thanks. And I'll be around if you have questions.